This video is going to help you to understand Punnett squares and how to work them through a little bit. Be sure that you go back and review this vocabulary: genes, alleles, chromosomes, meiosis. Understand how they all relate to the genetics unit that we've been addressing. Alleles can be dominant or recessive. Remember, capital letter is going to be dominant, recessive is lowercase letter. And then these are essential vocabulary words. Heterozygous is when you have the genotype, big A, little a. Homozygous dominant would be big A, big A. Homozygous recessive would be little a, little a. You've noticed that on a lot of the assignments that I've given, I don't refer to the genotypes. I simply refer to them as being homozygous dominant, homozygous recessive, heterozygous, any of those combinations. Remember, genotype, genetic makeup of the cell, that is just the letters. Phenotype is the physical trait expressed. Brown eyes would be an example. And then Punnett squares, the essentials of them is that they are a representation of the probability, just the probability of traits being passed on from two parents to one offspring. So when we have our Punnett square, remember that mom would be on the side here, and dad would be on the top of the square. And these possible squares are in here are the possible genetic outcomes. So let's practice filling in these squares. If you have parents of average height, mom is average height, dad is short. The genotypes would be big T, little t, the heterozygote with the homozygous recessive. Mom is big T, little t. Dad is little t, little t. You would pull these genotypes down and over. Here's another representation of that. So let's practice a mouse fur color. I asked you to cross a homozygous dominant, dad, with a homozygous recessive mom. I bring this B over, this B down. That makes it a heterozygote. This B down, this B over, heterozygote. Again, down, over, down, over. Our genotypic ratio would be 100% heterozygotes. And we can see that here because all of these within the boxes, the possible genotypes, are heterozygotes. Our phenotypic ratio would be 100% brown because the big B overpowers these little b's. Let's practice hitchhiker's thumb. Same kind of Punnett square. We've got a homozygous dominant with a heterozygous or a homozygous recessive. Again, down and over. Same genotypic ratio, 100% heterozygote. Phenotypic ratio would be 100% hitchhiker's thumb. We talked about these incomplete dominant rules. Remember that one allele in incomplete dominance does not completely dominate another one. Example, red would not completely dominate white. Instead, they would combine together to make pink. So let's practice one of these. Remember, I'm just putting my genotypes on the top and the bottom. And I wrote one incorrectly there. Let's do our genotypic ratio. Let's count them here. So there is 1 FR, 2 F big R, so it would be 2 F big R, F big R. Let me change that color. And then we would have 1 F big R, F little r, 1 F big R, F little r. So that's 2 heterozygotes. Phenotypic ratio. So we could also reduce that and make it 1 to 1. Phenotypic ratio. We would have 1, 2 red, 1, 2 pink. Again, 1 to 1. Codominance is an example when they have the red and white dots. Blood is also an example of this. Okay. Please go back and practice some more Punnett squares. Practice making up some genotypes, so you could just write some random letters to cross. Choose some letters. You can practice these that I'm writing if you want now. Just practice these as much as you can and think about them in terms of homozygous and heterozygous.